The main advantage of this technique is that it reflects RNA polymerase II activity by measuring newly synthesized mRNA, which are not influenced by RNA degradation. This protocol was originally described using microarray hybridization, but can easily be adapted to high-throughput sequencing of newly transcribed mRNAs. Demonstrating this procedure will be Tiago Baptista, a former grad student from our laboratory.
After inoculating the S. cerevisiae cells, grow them overnight at 30 degrees Celsius with constant agitation at 150 rpm. The next day, measure the optical density at 600 nanometers. Dilute the culture to an OD600 of approximately 0.1 in 100 milliliters of YPD medium. Grow up the cells until the OD600 is around 0.8. After this, measure the OD600 of the S-palm overnight culture. Dilute this culture to an OD600 of approximately 0.1 in 500 milliliters of YES medium, and let it grow up until the OD600 is approximately 0.8. First, prepare a fresh solution of 2 molar 4-thiouracil. Keep the prepared solution at room temperature and protected from light. Add the 4-thiouracil solution to the S. cerevisiae and S. palm cultures such that the final concentration is 5 millimolar.